Start recording, share my screen. So today in the lab, we will have a little bit of lecture with chapter 6, and then we will discuss lab 7, which I don't think I've got that open yet, but we'll get there. Oh, it looks like the other's done. That's good. So let's continue with the lecture. We talked about the effect oxygen has on microbial growth, and you can grow the organism in a thioglycolate tube to determine what the oxygen requirement for the organism is. Any question about any of that? If not, let's move on to talking about culture media. There are general purpose culture media and specific media. When we're talking about a culture medium, we're talking about one. And then when there's more than one, we use the word media. So media is plural, medium is singular. A culture medium is a liquid or solid solution containing all of the nutrients needed for growing microbial growth. In the lab, generally, you'll be given a sterile culture media. That is the one that has no living microbes in it. And how we get that is we autoclave it. The inoculum is the microbe introduced into the medium, and those would be the microbes you're trying to grow. The culture is the microbes growing in a culture medium. And so when you use the word culture, we're talking about the stuff you're growing, and it grows in a culture medium. There's a very wide variety of media that is available. And let me see if I can get a little book-like thing. If you were in the lab, I would hold up a book, and it's actually much bigger than this. It's probably about as thick as this. I don't know how tall. It's not even showing up well. Put that in the center. Black doesn't show up well. It's not showing up. Maybe against my white shirt, you can see it. There it is. So it's about that thick, the book, and maybe even thicker. And it's a bigger book too than this. We have a book having different media that you can grow your microorganisms in. And on the book, on every page of the book, is one media. So that tells you how many different media there are available for growing microorganisms and microbiology. A large number. Culture media are used to support microbial growth. The media must provide an energy source for the organisms to live, a carbon source, sources of nitrogen, sulfur, phosphorus, trace elements, and organic growth factors if the organism requires organic growth factors. The trace elements are usually obtained either from the water or like if you grind up the cow, the beef extracts from the cow, and the trace elements would be whatever the cow is eating and drinking. The energy source could be, if the only energy source is the beef extract, it could be ground up cow, and that would be the carbon source as well. And that could supply the nitrogen, the sulfur, and the phosphorus as well, because cows have all of that. But you could add these as molecules in the medium. Now that's for a liquid media. We also have agar, and other solidifying agents that we add to the liquid media to make them solid. Agar is a complex polysaccharide derived from seaweed, and we typically use agar in microbiology because microbes do not digest it. We could use starch or gelatin, but some microorganisms can digest those. So we typically use agar. Agar is usually used in a petri dish or in a test tube. In the tubes, there's two types. Let me see if I can get a picture. Oops, I need to do that. Where's the, oh, there's nothing in the window. So of a test tube, we can have the agar going straight across, and that we call a deep. You can also have the agar growing on a slant. Oops, let me see if I can erase that. There we go, that's better. In which case you grow the bacteria on the slant. And it's possible you could have both. Let's make this one wide. Where we have agar first as a slant, as a deep, and then on top of the deep, you have the slant. Or you can have it there, yeah, not there. So it'd be like that. Anyways, those are different test tubes. A deep, a slant, and that's a combination. Agar is generally not metabolized, and that's why it's used as a solidifying agent. You can have different types of media. There's chemically defined media, where the exact chemical composition of the media is known. All of the molecules in the media you added to make the media. So here would be a simple one. Let me see if I can blow that up. So this one has glucose, ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, and then water. These are the only molecules in this media. Assuming we're using distilled water, there would not be other molecules getting there other than maybe oxygen and carbon dioxide from the air. Everyone understand the chemically defined media. You know all of the molecules in the media. These chemically defined media are usually used for autotrophs because they are simpler to grow. You just need a carbon source, glucose, an energy source, glucose, a phosphate source. This is the correct, we make the correct osmolality. This is the sulfur source, the magnesium source, and the phosphate source. And then everything else for the autotroph you can get from the air. Any question about that? Now, a heterotroph would not grow in this because you're not supplying enough organisms, enough elements and nutrients. That's the word I want to use, nutrients. Like for example, there's no copper source and no magnesium source. Well, actually there is a magnesium source. No copper source, no iron source. If the organism required vitamin C, there's no vitamin C source. If many growth factors are required by the organism to grow, we generally call that a fastidious organism. And here, this media is supplying something. Let me see if I can draw that up. Something for a fastidious organism. All the different things we're adding. There's phosphate, sodium chloride, another phosphate, bicarbonate, I don't know what that is added, potassium sulfate, so potassium and sulfur, etc. And then the organic growth factors, calcium panthenate, thiamine, nicotinamide, adenine dinucleotide, uracil, biotin. These are vitamins, hypoxanthine, sodium thioglycolate to remove the oxygen, cysteine for an amino acid. So this organism is fastidious. Now, most fastidious organisms would not grow here because they require other things like vitamin C. We'll talk about fastidious organisms in a minute. But let me talk about a complex media. This contains an extract of digestive yeasts, meats, or plants. The digestive yeast, meats, or plants provide vitamins and organic growth factors. The exact chemical composition of these media are not known, and they will vary. For example, this one here, we're adding peptone, that's a digestive protein from soybeans, beef extract, so we ground up a cow. Well, this beef extract will have many different molecules in it. Whatever the cow was eating, the cow will have those molecules, and then that will go into the complex media. And usually we put sodium chloride to make the osmotic pressure correct, and then agar to make it a solid media. If it's a broth, a liquid media, we wouldn't add agar. The peptone is a protein digest. It's the primary energy source, the source of carbon, nitrogen, and sulfur. But the beef extract can also be an energy source, a source of carbon, nitrogen, and sulfur. We use complex media to grow heterotrophs, bacteria and fungi that require an organic chemical carbon source. Like E. coli would be a heterotroph, and so we grow it on a complex media. So that's a complex media. There's also anaerobic media. An anaerobic media is one that we use for growing obligate anaerobes. These are organisms that must avoid exposure to oxygen. And anaerobic media can also be called a reducing media because it has a chemical that reduces, like sodium thioglycolate, that combines and removes oxygen from the media. And so the reducing media will be oxygen depleted, at least at the bottom of the tube. And you heat that media before using to drive off any absorbed oxygen from the media. There's also selective media. A selective media is one that has chemicals which halt or inhibit the growth of a particular group of microorganisms, but it does not affect the growth of another group of microorganisms. For example, 7% sodium chloride media is a selective media because it contains 7% sodium chloride. If the organism can grow in 7% sodium chloride, it will be selected for. If the organism cannot grow in 7% sodium chloride, that organism will be selected against. Like E. coli cannot grow on 7% sodium chloride, and Staphylococcus aureus can. Any questions about any of that? All right, so that's the selective media. There's a differential media. A differential media provides differences that you can see between two groups of organisms. The differences are not related to how well the organisms grow on the media. The differential media makes it easy to distinguish colonies of different microbes because they look different. And they could have different colors, they could do different things, but they provide two species to show differences. For example, blood agar is a differential media. That doesn't work there. I have to put it there. With blood agar, we can look at or differentiate between the different types of hemolytic activity the cells have. You can have, you can have non-hemolytic organisms where the blood of the plate is not lysed. Come on, mouse. When we come and see the blood, it's red all the way up to where the bacteria is. This is no hemolysis, which is called gamma hemolytic activity. On the other hand, you can have alpha hemolysis, which is partial hemolysis. And as you can see, the blood, it's red. And then right before we get to the growth, which is white or off-white, we have a dark ring surrounding the growth. And that is alpha hemolysis, partial hemolysis, where the bacteria are secreting something that is partially lysing the red blood cells. And it can look either dark or greenish. And if you notice inside the growth here, inside this alpha, that's a dark alpha. And that's because we have partial hemolysis everywhere here. Any questions? You can also see complete hemolysis. And here we have the bacteria, which is white in the shape of a B or a beta. And around it, we have the agar with the blood completely lysed. And that's why it looks clear. And that we call beta hemolysis or complete hemolysis. Any question about that? Beta hemolysis, alpha hemolysis, gamma hemolysis. You'll note that we have a dark ring around the beta hemolysis. This is actually a ring of alpha hemolysis, and it is normally seen wherever you have beta hemolysis, you'll have further out alpha hemolysis. Anyways, that's blood agar, a differential media, showing you differences between different types of cells, one that has beta hemolysis, one that has alpha hemolysis, one that has gamma hemolysis. There's also selective and differential media. It is both selective and differential. Eosine methylene blue, or EMB plates, are an example of the selective and differential. It is selective because it selects against gram-positive bacteria. It contains eosine and methylene blue, two dyes which get into the cell walls of the gram-positive cells and then prevent the cells from growing. Meaning the cell doesn't like those two dyes. It gets into the cell walls and interferes with the cell wall, preventing them from growing. And so the gram-positives cannot grow, and the gram-negatives can grow. So it selects for gram-negatives, selects against gram-positives. It's also differential on the basis of lactose fermentation. If the organism can ferment the sugar lactose, and obviously if it grows on the plate, then it will look dark like this or a metallic green like that. And that's from the fermentation of the sugar lactose, changing the pH, making it more acidic, and it looks metallic or dark when it is a lower pH or acidic. If it can grow on the plate, but it does not ferment the sugar or lactose, then it will not be dark or metallic. And as you can see, these colonies look white, and that's because this organism cannot ferment the sugar or lactose. This organism can, so it's dark and metallic looking. And then they're both gram-negatives because they're growing on the EMD plate. Any question about selective and differential media? We also have an enrichment media, and this media encourages the growth of certain microorganisms and selects against other microorganisms. Therefore, it is a selective media, but it's not used in the way a selective media is normally used. We use an enrichment media to encourage the growth of the organism of interest. So when we have a population of microbes that we want to study, and the number is very rare, like you're looking at a microorganism from the soil, and it's very rare in the soil, or you're looking at a potential human pathogen in fecal matter, and it's a rare microorganism, well, we can grow it in an enrichment media and enrich for the organism of interest. And if you enrich enough, you will then be able to find the organism when you streak it out. For example, let's say we're interested in the soil organism that degrades phenol. And there's thousands of other bacteria in the soil. So to streak it out on a plate, you might get 10 colonies. If only 1 in 1,000 is going to be the organism of interest, you're not going to be able to find the organism of interest. You would need to streak out 1,000 colonies just to get 1. Everyone see that? So what you can do is grow it in an enrichment media. Let me see if I can grow my plate up again. Let's do that again. You can have the enrichment media and inoculate it with the soil bacteria, and it'll increase the number of microbes. So instead of 1 in 1,000, we can increase it to 1 in 100. You'd still have a hard time finding your organism of interest if it's 1 in 100. That means you'd have to streak out 100 different colonies. That's not very good. But we could then take from the, see if I can get that down. Take from this and then take an aliquot and put it into another tube. I'll put it here. And grow it again. And now we can increase the microbe from 1 in 100 to 1 in 1,000. Not 1 in 1,000, 1 in 10. So if it's 1 in 10 colonies, we could easily streak out the cells, and we'd be able to obtain the organ of interest, because we only have to test for 10 colonies, and we're likely to find 1, which is the organism of interest, in this case, phenol degrading organism. Any question about any of that? Well, how can we use an enrichment media? Well, if we're interested in a phenol degrading microorganism, what we can do is create a media where the only carbon source is phenol. And then the only organisms that can grow in this media are organisms that can utilize phenol. And so that will select for the organism of interest. and select against the organisms that we're not interested in. All right. Any questions about any of that? And if that wasn't enough, you could also take from, you could take from this one and do it again. I'll have to go like that. So you can enrich more than one time. All right, any questions about enrichment media? Any questions about media in general? This table is showing you the different types, the summary table, chemically defined media used for growing autotrophs. You know all the molecules in it. The complex media, the growth of most heterotrophs, the reducing media, for growing obligate anaerobes, a selective media, where you're selecting for certain organisms and selecting against other organisms, differential, allowing you to see differences of the two different colonies, two different organisms growing on the plates, and then an enrichment media, similar to selection, but designed to increase the numbers of the desired microorganisms to detectable levels. All right, any question about any of that? If not, I think we'll stop here. Let's see if I can put that in there. All right, let's go to the next slide. We'll start here. All right, there's no questions. This is it for the lecture. Let's go to the lab. Lab 7, if I remember. Oh, I got it here. There is some pre-reading that you should do before you do the lab. Let's put that on the bottom. The learning objectives, be able to explain how to properly collect throat and urine specimens for routine culture, be able to list common bacterial pathogens isolated from the throat and urine cultures, know how to culture media and how to cultivate these organisms, they know the culture media you use to cultivate these organisms, recognize which tests are used to identify throat and urine pathogens, and understand the principles of how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. Any question about what we're doing? So once a specimen is received in the lab, they're logged and processed in a timely fashion. Specific culture media are inoculated and incubated for a prescribed length of time. And we use different mediums for throat and urine specimens. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. So if we have a throat specimen, we grow most of them on blood agar, and some of them on chocolate agar. For throat specimens, the most common throat pathogen is Streptococcus pyogenes, and it grows on blood agar. But especially in children and babies, sometimes we can get Haemophilus influenzae in the throat, and that will not grow on chocolate agar. Excuse me, not grow on blood agar. We have to grow it on chocolate agar. Any questions about any of that? All right, for urine culture, we normally grow urine cultures on two different media. One would be blood agar, the other would be either MacConkey's agar or E&B. For blood agar, you're going to grow both the gram positives and the gram negatives, as well as anything that's finicky in growing. The term is fastidious. And then MacConkey's and E&B agar, we grow it on these two media, because usually in the urine, if the human has a pathogen, usually most of the human pathogens in the urine will be from the Enterobacteriaceae family. And so we want to grow it on MacConkey and E and B because they will grow on these media. The gram-positives will not. And here are some common bacterial pathogens that you can find from a urine specimen. Streptococcus is a gram-positive. Enterococcus, I think that's gram-positive. That one I have to look up. Staphylococcus, gram-positive. And then Enterobacteriaceae, as well as the Pseudomonaceae. The most common bacteria that causes a urinary tract infection is actually E. coli, which is an Enterobacteriaceae. All right, any question about any of that? All right, so sore throats may be caused by viruses or bacteria. If you have white patches on the inflamed tonsils in the back of the throat, there's an indicator of a bacterial infection. If it is not white patches in the throat, then that's indicative of a viral infection. And a clinician does look at the throat to try and determine is it viral in origin or bacterial in origin. If it's bacterial, you can treat it with antibiotics. If it's viral, you should tell the patient, essentially try and get lots of rest and drink lots of fluids. There's not, well, there's some antiviral drugs, but they usually don't prescribe antiviral drugs unless the virus infection is very severe. The most commonly isolated bacterial pathogen from the throat is Streptococcus pyogenes, and it's also called Group A strep, which I don't use that term. Or Group A Streptococcus. So I list the species Streptococcus pyogenes. If you have strep throat, what you want to do is swab the back of the throat or the tonsils where there's redness and white patches. You do not want to touch the sides of the mouth and the tongue. Patients that test positive in a blood agar plate, you want to determine whether they're positive for Streptococcus pyogenes, because if they are, you want to put them on antibiotic therapy. Streptococcus pyogenes is difficult for the body to throw off, and that's why oftentimes when people get strep throat, they can't throw it off. It's because the bacteria have a capsule, and we'll talk a little bit more about that. So Streptococcus pyogenes is a serious human pathogen. It's also fastidious, and it will not grow on a tryptic soy agar or a general purpose media plate. You have to grow it on a enriched media, such as tryptic soy agar, supplemented with 5% sheep's red blood cell. The blood supplies the nutrients for Streptococcus pyogenes to grow. And then what you look is on the blood agar, if you're looking for Streptococcus pyogenes, you look for small colonies that are surrounded by a complete zone of hemolysis or beta hemolysis. However, there are other beta hemolytic bacteria, even beta hemolytic streptococcus, and so you need further tests to prove that it's Streptococcus pyogenes. You only need to do 2 biochemical tests after you find beta hemolysis on a blood agar plate. You do the catalase test and the basic trace and susceptibility test. Remember I told you you do the catalase test. Let's look at blood agar here. Oops, that didn't blow up. So this is normal throat, where this is no hemolysis, or where it's dark here, there's partial hemolysis. And this right here is beta hemolysis, where we have the bacteria, which is the white circle in the right there, and it's surrounded by a zone of complete hemolysis, which is very clear. So there's the bacteria, and there's the clear hemolysis of the blood. Any question about any of that? To do the catalase test, you just look for the enzyme catalase. Most bacteria that grow in oxygen can produce catalase. Catalase takes hydrogen peroxide and makes water and oxygen. The oxygen being produced produces bubbles in the water and the hydrogen peroxide. And so you look for the production of bubbles, as here. And if you have bubbles, then the organism is positive for catalase. If there's no bubbles, then the organism is negative for catalase. If the organism is negative for catalase, why is it growing in oxygen? How come it can grow in oxygen? Does anyone remember? It has no catalase. Streptococcus pyogenes is growing in the air, well, exposed to the air, and it has no catalase, so it'll be catalase negative. So how is it able to grow in the air? Does anyone remember? It has peroxidase, which converts the hydrogen peroxide into water. So most bacteria growing in the air will have catalase. Our skin has catalase, but some bacteria have peroxidase, and it will remove the hydrogen peroxide, so it won't be toxic to the cells. All right, so like I said, Streptococcus pyogenes will be catalase negative. And then we do the bacitracin susceptibility testing. You plate the bacteria over the plate. So this whole region has been given the bacteria. And then you put a disc containing bacitracin, that's an antibiotic, on the growth. And if you get a clear zone of inhibition where the bacteria cannot grow within that, and the zone is large enough, we're not going to talk about that here, but we will later, then it's susceptible to bacitracin. The Streptococcus pyogenes is susceptible to bacitracin. So group A, Streptococcus pyogenes. So this organism, Streptococcus pyogenes, this beta hemolytic Streptococcus is not sensitive to bacitracin. So it is not Streptococcus pyogenes, it's some other member of the Streptococcus family. All right, another organism that can clinically be significant is Haemophilus influenzae, gram-negative Coccobacillus. It's commonly seen in eye, ear, and in sinus and in throat infections in babies and young children. It's a fastidious organism, but it cannot grow on blood agar. But it can grow on chocolate agar. So Haemophilus influenzae growing on chocolate agar, right here. What chocolate agar is blood agar where the blood was added when the agar is still very hot. And it's hot enough to lyse the blood. And that makes the agar turn brown, looking like chocolate. That's why they call it chocolate agar. Anyway, Haemophilus influenzae can grow on chocolate agar. When we streak Haemophilus influenzae here, there's no growth because it cannot lyse the red blood cells. And if it doesn't lyse the red blood cells, you can't get the nutrients from the red blood cells. So Haemophilus influenzae cannot grow on blood agar unless you put a streak of Staphylococcus aureus on the blood agar and you streak the Haemophilus influenzae parallel to it. And then the Haemophilus influenzae can grow in this region here around the Staphylococcus aureus, because the Staphylococcus aureus lyses the red blood cells, and that will release the nutrients for Haemophilus influenzae to grow around Staphylococcus aureus. But further away from Staphylococcus aureus, we see no growth. Any question about any of that? All right, let's move on to urine cultures. Urine cultures are used to diagnose urinary tract infections or UTIs. Bacteria can travel to the bladder through the urethra. Because females have a shorter urethra than males, they are predisposed to getting bladder infections. Now, normally, the urine flowing through the urethra washes the bacteria out of the urethra, so it does not get up to the bladder. But every once in a while, the bacteria can cause a bladder infection, and it's more likely to be seen in females than in males. When you're collecting the urine from a patient, there are 3 ways to collect. Let's see a picture of the first way, other than here. The best way to do it, and the way the patient prefers, is the clean catch midstream, where you swab the urethra, and then you let the urine flow for a little bit before you collect it in the container, and then you collect the urine midway through the urine, and then you stop, cap it. Obviously, you continue urinating, but the point is you catch midstream in the urine, and that reduces the number of contaminating bacteria coming from the urethra. But there will be bacteria coming from the urethra in the clean catch midstream. Another way to get the urine sample is to put a catheter up the urethra and into the bladder, and then sample the urine from the bladder. This is not perfect because when you put the catheter up through the urethra, the catheter can pick up bacteria and then deposit them in the bladder. But the bacteria you're getting is much reduced from a clean catch midstream. And then the third way, the suprapubic aspiration, is you put an injection directly into the bladder and then sample the urine directly from the bladder. Now, this way, patients definitely wouldn't like that because you're poking them with a needle. And you have to be skilled at doing that, meaning they usually will be a little prepared. I'm not sure how easy that is to do because I've never done it. Anyways, if you do a suprapubic aspiration, you should not have any contaminating bacteria. So any bacteria you get out of that should be the human pathogen causing the urinary tract infection in the bladder. So once the specimen is received in the lab, you only plate 1/1000th of a milliliter of the urine onto a non-selective media, such as blood agar plate. And I actually have a specific tool for doing that. This tool here is for putting urine, streaking it on the plate, showing you how you streak it on the plate using this swab. And that little swab there has a volume of exactly 1/1000th of a milliliter. And that's the size of the loop compared to the size of a penny. Any question about that? And the Canadian penny is the same size as our penny. All right, so we normally inoculate blood agar plate and also a selective media, either MacConkey's or eosine methylene blue, and that's to grow specifically coliforms such as E. coli, the most common isolate in a urinary tract infection. We call coliforms, never mind, you can read about that. The interpretation for positive urine culture is multifaceted. Several factors have to be considered. The type of collection, for example, midstream clean catch system, as opposed to a catheter or opposed to a supra pubic aspiration, will have different numbers of contaminating bacteria. The supra pubic aspiration should have no contaminating bacteria. The catheter, only a few, and then the clean catch midstream, more. The gender of the patient will determine how many bacteria in the urine, meaning a female will have more bacteria from the urethra in the urine than a male will. The number of colonies per mil in the urine, generally, the more colonies per mil you have in the urine, the more likely you have a human pathogen. And whether there's a presence of polymicrobial growth, meaning if there's many different organisms growing on the blood agar and then the E&B or the MacConkey's, it's a good bet if there's many different bacteria species growing, and there's probably a pathogen being one of them. All right, there are a few tests we do once we have the bacteria growing up from a urine culture. The first is the coagulase test. Coagulase looks for the enzyme coagulase, which can be used to differentiate Staphylococcus aureus from other Staphylococcus species. Staphylococcus aureus is coagulase positive. The other Staphylococcus species are coagulase negative. Coagulase is actually a factor increasing the pathogenicity of Staphylococcus aureus, because if the organism can produce coagulase, it will cause the blood plasma fibrinogen to change into fibrin, which will start the blood to clot. This will actually protect the Staphylococcus aureus when the bacteria infection are low in number, because you get a clot around the bacteria. It's harder for the immune system to get in and kill the bacteria. So the clot is something that Staphylococcus aureus uses to help protect the bacteria from the immune system, largely preventing or helping prevent phagocytosis. Any question about any of that? All right, there are two coagulase tests. The one I'll use in the unknown project is the clot forming test. And there's also the agglutination test. In the agglutination test, you put serum, and then you add the bacteria, If no clotting happens, it's coagulase negative. If clotting or clumping happens, it is coagulase positive. You can also have a serum, a tube of serum, and if the organism, you put the organism in the serum, if the organism does not produce the enzyme coagulase, the serum will stay liquid. And that's why when you slant it, the serum runs. Or if the organism is coagulase positive, the coagulase will clump the serum, forming the clot, and that you'll see like that. So this is coagulase positive. All right, any questions about coagulase? The most common bacteria isolated from urine culture is E. coli. This is a member of the family Enterobacteriaceae, which is a gram-negative bacilli. Another gram-negative bacilli that can be a urine pathogen is Pseudomonas aeruginosa from the Pseudomonaceae family. It is a gram-negative bacillus, but it's different from the Enterobacteriaceae because instead of being facultative anaerobe, it's an aerobe. Instead of fermenting the glucose, this one does not ferment glucose. And then this one will be oxidase negative, and then Pseudomonaceae is oxidase positive. Oxidase positive is an organism that has the enzyme cytochrome C, so that's what we're looking for. And in the oxidase test, the reagents will turn purple in the presence of the enzyme oxidase. So the Enterobacteriaceae will be oxidase negative, meaning they'll have no color. And then the Pseudomonas aeruginosa, as well as any Pseudomonaceae, will be oxidase positive, and they will be turning purple from the oxidase test. All right, so there's a summary you can read. You don't need to do anything with the references. And then let's talk about the exercises. You're not physically going to be performing any of these activities. However, you will be responsible for knowing how to perform these activities. We have a video clip on the catalase test. We had a virtual interactive laboratory exercise, but we decided to scratch it because it's difficult to use. It requires the use of Flash, and Adobe has turned off Flash on all computers now. So Flash will not work, and we're not going to have you download a Flash, what do you call it, program, and then you open up the file directly in that program, and then you could watch the interactive laboratory. And we decided it was too difficult, so we won't be doing that. You can watch a video clip of the coagulase test and a video clip of the oxidase test. So watch these videos and then work on your questions for the exercise. Remember, when you're working on your questions, don't work on them in the lab module. Work on them in the lab worksheet, which I'm trying to show you here. You can't close that. That reminds me, I've got to put that on the video on YouTube. Are we on lab 7 or lab 8? I think we're on lab 7. So do your lab exercises, which you can find from a link in Canvas. So scroll down to the appropriate week. sure that's turned on. What week are we on? There it is, Worksheet Lab 7. So click here to get the worksheet and then answer your questions in the laboratory worksheet. It's similar, except you'll only have the video clip. And then answer the questions, and let's see if there's anything specific. You do not need to do question 8C. That is not discussed in the lab module. All right, any questions? If not, I'll be here until 8 o'clock. So I'll be here for half an hour to answer questions. Generally, students will log off early, but I'll be here until 8 o'clock. The point is, if you have a question, make sure you log in before 8 o'clock. All right, start working on the lab, and that's it for the video. I will be working on sending you the unknown project.